This is 200, 500, okay. Okay. And we're going to say, and 200 millimolar sucrose here. So, and we're going to start with, at equilibrium, we have the same volume, the same level in tank A and B. Okay, we're going to start with that. And now the permeability, we're going to have relative permeability. So I'm going to write relative permeability here. And we're going to, thank you, we're going to say that the permeability to water is higher. So we're going to say that the permeability to water is going to be 1,000. We're going to say that permeability of urea, permeability of sucrose, sorry, not glucose, but sucrose, and permeability sodium chloride, permeability calcium chloride. So here we're going to say okay. So these are the relative permeabilities, okay, of that membrane X. So first thing, what do we do? What do we do? And give the same example. Convert everything to milliosmolar. So 300 millimolar urea is going to be, what is the osmolarity? The same, right? 300 milliosmolar, because it's going to be dissociation number of 1, right? How about sodium chloride? 100 millimolar is going to be what in osmolarity? 200, because sodium chloride dissociates into sodium and chloride, so a dissociation number of 2 gives us 200 milliosmolar. How about sucrose? What's that? 200 milliosmolar. Sucrose does not dissociate. What about calcium chloride? What's that? 300. Calcium chloride dissociates into 1 calcium, 2 chlorides. That's 3 solute particles. So dissociation number is going to be 3 times 100. We're going to have 300 milliosmolar calcium chloride. What is the total osmolarity of tank A right now? $500, right? So we add these two. This is 500 milliosmolar. What about tank B? Total osmolarity of tank B, 500. So my first question to you guys is tank A or compartment A isosmotic, hyposmotic, or hyper? It's not letting me write down here. I'm going to write it here. Hyperosmotic to B. Before any water crosses. Before water crosses. What do you guys think? Isosmotic. Same osmolarity here, same osmolarity here, before anything is crossing at time 0. Everybody understands that? Yes? Somebody doesn't? I can explain it again. So here, before any water crosses, water is the one with the highest permeability. If water hasn't started crossing, nothing else has started crossing. Okay, so you're at time 0. And at this point, we look at just the osmolarity of tank A and tank B, and it's the same, so it's isosmotic. So both tanks have the exact same osmolarity before anything crosses. So isosmotic, okay? So now I'm going to ask you a second question. is A going to be isotonic, hypotonic, or hypertonic as water starts crossing? So now water starts crossing, other things are going to follow, start crossing as well. So at this point, what we want to do is look at what is osmotically active and what is osmotically inactive, okay? So we're going to, let me change my ink color here. So we're going to look at what the membrane is permeable to, all right? So water is going to start crossing at this time, time 1. All right, so water starts crossing is the first thing that is going to start crossing because it has the highest permeability. Is water going to move in one direction relative to the other one, from compartment A to B or B to A? No, right? Because it's 500 millismolar, 500 millismolar, so there's going to be water crossing, but not net movement of water. So at time 1, nothing is really moving because both compartment A and B are isosmotic. Then things are going to start moving. And so let's look at urea first. Urea, the membrane is permeable to it, right? So urea is going to move from A to B or B to A. And I'm going to erase this here so I can. So what do you guys think? Urea. Urea is going to move from A to B, right? Because urea, we have some urea in A, but nothing in B, so it's going to move down its own concentration gradient until it equilibrates, both tanks equilibrate. So just to give you guys numbers, it was 300 here, so let's say we're going to do 150 urea here and 150 urea here, okay? Actually, let me put it up here. What about glucose? No, sucrose, sorry, sucrose. Can sucrose move? Yes or no? Yes, right? We have a driving force and we have a pathway. The membrane is permeable to sucrose, and we have sucrose in B, but no sucrose in A. So sucrose is going to move from B to A, okay? And they move sucrose and urea at about the same rate, right? Because the relative permeability is 100. So we had 200 millismolar, so we're going to have 100 here, right? It was 200 millismolar, right? Yeah, so 100 and 100. So it moves from B to A until it equilibrates. What about sodium chloride? And I should put here sucrose, sorry. Sodium chloride will move. Oh my God, hold on. I messed up. What was I took a picture of this. What was I forgot how many. Okay, we had 100 milliosmolar sodium chloride. So before I move on here, just because I erased my 100 here. So 100 sucrose, 100 sucrose, right? So we're still there. Okay, we're good. Thank goodness I took a picture of this. Now 100 milliosmolar sodium chloride. So we have sodium chloride in A, we don't have sodium chloride in B. So that means that we have a driving force. And the membrane is permeable. It's not 0, it's 1. Permeability is low, but it's a relative number, right? So it's 1,000 times less permeable to sodium chloride than it is to water, but it's still permeable. So sodium chloride is going to start moving down its own concentration gradient towards B, okay? So sodium chloride moves towards B. We're going to end up with 50 sodium chloride here and 50 sodium chloride here, okay? How about calcium chloride? Calcium chloride cannot move. We do have a concentration gradient trying to move it from B to A, but the permeability is 0. So calcium chloride remains there. So we add up the permeability, the osmolarity, sorry. So we have 150 urea, 100 milliosmolar sucrose, and 50 sodium chloride. That's going to be 300 milliosmolar here. And in B, we have 100 sucrose, we have 150 urea, 50 sodium chloride, so that gives us 300 again, plus 300 of calcium chloride, right? So that's going to be 600 milliosmolar, right? Did I do the math correctly? Yeah, 200, 300, 600, right? So now that we have 600 milliosmolar here and 300 milliosmolar here, after some time of these osmotically active solutes crossing the membrane and the osmotically inactive solute, which is calcium chloride, not being able to move, what is going to happen to water? Is water going to keep moving from, it's going to move from A to B or B to A now? What do you guys think? A to B. So water is going to move from A to B. Okay? If that is the case, if water is moving to compartment B from A to B, then A is what? Isotonic, hypotonic, or hypertonic? Hypotonic. This one now has the higher osmolarity. So this one, B, is hypertonic. A is hypotonic as water starts to cross. Does that make sense? Yeah? So that's how you solve these problems, right? First, you look at the osmolarity of both. You convert, first step, you convert everything to milliosmolar, okay? Once you have that, you look at the total osmolarity of one compartment relative to the other one. Before any water crosses, I'm always going to give you water crossing more, the membrane being more permeable to water. I'm always going to give you relative permeability. So you can compare the permeabilities fairly easy. You just look at the numbers before any water crosses and you determine, okay, is the osmolarity in compartment A higher, the same, or lower than B? And you can determine if it's iso, hyper, or hypoosmotic. As water starts to cross, then you're going to look at what water is going to do. In this case, it was isosmotic. So water, there was no net movement of water. But then the solutes will start moving. So you look at, do we have a concentration gradient or osmotic gradient, and do we have a pathway? And if we have those two things, then that particular chemical can move. Okay? So in this case, urea, sodium chloride, and sucrose could move. They're osmotically active. The membrane is permeable to them. And we have a driving force. Okay? And calcium chloride is osmotically inactive. Even though we have a driving force, we don't have a pathway. The membrane is not permeable to it, so it cannot move. Okay? In that case, then you end up with a higher osmolarity in compartment B because the other three solutes are going to equilibrate, right? But the osmotically inactive solute remains in that compartment B. So at this point, water is going to move to where there's higher osmolarity, in this case, compartment B. So we know that A is hypotonic to B, or you can say that B is hypertonic to A. Does that make sense? Tonicity is going to refer to the movement of water given the osmotically active and inactive solutes. All right. Now I'm going to talk about the kidneys, okay? And the function of the kidneys, sorry, before I move on, everybody's good? Yeah. The concept is different, yes. Yeah, because if the solutions are isotonic, then you're going to have, they're going to be isosmotic. Yeah, because the water is not going to move. So tonicity, so the concept of tonicity is relative to the water movement given the osmolarity. Does that make sense? So they are the same. They're both going to, the water movement is going to be determined by the osmolarity. It's just you can measure an osmolarity, just comparing the numbers. Tonicity refers to the movement of water given what solutes can move or not move. If it was a system in which everything can move, then it's the same osmolarity and tonicity is going to be the same. But because you have osmotically inactive solutes, things that cannot move either because you don't have the driving force or you don't have the pathway, then tonicity will be different than osmolarity. Having said that, you can also say now at that time, whatever, time 5, right, when we reach equilibrium, that B is hyperosmotic than A. But I'm not asking you for that. If I were to ask you, okay, what is A to B in terms of osmolarity? Iso, hyper, or hyposmotic at time 5, at the end, when all the solids move as well. So as water starts to cross and all the solids, what you would say that A is hyposmotic and hypotonic. Does that make sense? But at the beginning, when I asked you before any water crosses, you cannot talk about tonicity because tonicity involves the movement of water given the solids. Do you know what I mean? So here you can talk about tonicity and osmolarity when we're back at equilibrium at the end. But but B is now B is hypertonic, and that's what caused that movement of water. Until you reach equilibrium, right? So now the volume in B is going to be higher than in A because water moved. But that was because tank B is hypertonic to A, and you reach that new equilibrium. So the volumes are now different. B gained gain water. So volume in B is going to be higher.
And then at that point, they would both be isotonic and isosmotic.
At this point, the osmolarity is different, right? So 300 and 600. We reached that new equilibrium with B being hyperosmotic. It's just the volume is higher. Does that make sense? The number of solid particles per volume. Okay, so I see what you mean. Okay, I see what you mean. Yes, you still have 600 milliosmolar, right? Because you have a higher number of solid particles, right? But water moved to try to dilute that. So you did increase the volume of water. So yeah, eventually that osmolarity will be the same. Yeah. Yeah, I know, it is mind-bending, and it is tonicity what is mind-bending because it's such a abstract concept. It just refers to the movement of water. So at some point, this dynamic will reach equilibrium again because a bunch of water moved and made the osmolarities, equilibrated the osmolarities, but the volume in B is going to be higher than in A. Is that what you were getting at? So yeah, so at the end, the osmolarity would be the same because water will dilute that number of solid particles in B. Is that what you're asking me? So then they'll be isosmotic again because you're dividing by a larger volume. Yeah? Okay, good, no problem. That was a great question, by the way. Oops, I dropped the microphone. Okay, so we're going to move to kidneys now. And so for kidneys, I need to talk to you guys about the function of the kidneys, which normally you think about excretion being the function, producing urine and excreting a bunch of stuff in that urine. But that's not the only function of the kidneys. Kidneys are going to regulate extracellular fluid, volume and blood volume, okay? So the main extracellular fluid compartment is going to be plasma, okay? There's a number of extracellular fluid compartments, but plasma is the biggest one, okay? So your blood really is the biggest one. And the kidneys can regulate the volume of this extracellular fluid by either conserving water, so instead of excreting all that water in the urine, they're going to conserve that water and recycle it, bring it back to the circulatory system. Or if you have too much water in that compartment, then the kidneys would excrete a large volume of urine. And so, given that the main extracellular compartment is plasma, it can regulate blood volume. And if we regulate blood volume, we regulate blood pressure. Because remember, blood pressure is a hydrostatic pressure. It's the pressure that the blood exerts against the walls of the blood vessels, right? So by regulating blood volume, we regulate blood pressure. It regulates ion levels in the blood, so things like sodium, potassium, calcium, it's going to be regulated by the kidneys. So in the last part of the last segments of the nephron, we're going, and as I'll show you that later today and on Friday, these ions can be regulated. If you have too much sodium, then you're going to remove that sodium. If you have too much potassium, you remove that potassium. And this is really important because maintaining the levels of sodium, potassium, and calcium in the extracellular compartment, in that blood, is important to maintain the resting membrane potentials, sodium and potassium, remember, the resting membrane potentials of cells that will determine how easy it is for these cells to produce an action potential. And also, levels of calcium are important because, remember, calcium is the signal for contraction, and it's also a cellular signal in a number of signal transduction pathways. So regulating ion levels in the blood is super important. Homeostatic regulation of pH. We talked about pH regulation with the respiratory system. The kidneys also regulate pH. If your blood is acidotic, then the kidneys are going to excrete hydrogen ions and reabsorb bicarbonate, bring bicarbonate back to the circulatory system. If your blood is alkalotic, the pH is too high, then the kidneys excrete bicarbonate and reabsorb hydrogen ions. So they can control acid base as well. And then excretion of wastes in urine, things like ammonia, urea, uric acid, bilirubin, which is a byproduct produced when you break down hemoglobin in red blood cells, and then hormones are also excreted in the urine. And it also produces hormones. So it produces erythropoietin, which is a hormone that goes to the bone marrow and triggers or initiates erythropoiesis, which is production of red blood cells. So it participates in increasing the oxygen carrying capacity of the blood. Red blood cells are the ones that have hemoglobin, the ones that, the cells that are going to carry oxygen to the tissues bound to that hemoglobin and remove the CO2, CO2 also bound to that hemoglobin. Calcitrol is the active form of vitamin D, so it's important for your bones. And then renin, it's an enzyme produced by cells in the kidneys, and we'll talk about that later on, that helps in regulating blood pressure. Okay, helps in the production of angiotensin II, which is a hormone that regulates blood pressure and a ton of different things in your body. Super important hormone, angiotensin II. Okay, so let's look at the anatomy of the kidneys first. The kidneys are located on your lower back, so the lumbar region, but on the back, so behind all the other viscera, all the other organs. You know, when you have pain right here, they normally look at your kidneys. Could be back, muscle pain, but sometimes if you have other symptoms, they figure out it's a kidney infection or something like that. So normally the kidneys are back here, lumbar region in the dorsal part. Here's the kidney, so you have two kidneys. Here is one of the kidneys showing the two layers. We have two layers, the renal cortex. Renal cortex, as the cortex of a tree, is the outside of the kidney, right here. And then the renal medulla is going to be the middle part. Here I'm going to zoom in this segment of the kidney. You have here the renal cortex, and here the renal medulla. And you see these yellow squiggly things here? These are nephrons. Nephrons are the functional unit of the kidneys. They're the ones that are going to produce urine. And you have some parts of those nephrons in the renal cortex and some part of those nephrons in the renal medulla. Here you have the renal pelvis. And so at the end, these parts of the nephron in the renal medulla that made the urine are going to dump that urine in the renal pelvis. And from the renal pelvis, it goes to the ureter. You have two ureters connecting each kidney to the bladder. In the bladder, the urine is stored, and when need be, you go and empty the bladder. Okay, so your urine is going to go out the urethra to the environment. So being inside the nephron is considered being outside your body. Same as digestive system. Everything that is in your intestine is considered outside the body. The fluid in the nephron considered outside the body. Okay? So just that concept is important for a little bit later to see how things work. Now, the kidneys work really closely together with the circulatory system. Okay? Because the circulatory system is what's going to bring things towards the kidneys to excrete. Sometimes the kidneys are going to reabsorb that, recycle it, bring it back to the circulatory system so that we don't waste it. Sometimes things that didn't get filtered initially are going to be secreted into the nephron again to be excreted. And I'll show you how that process works in a minute. Let's take a closer look at this nephron here. So you see how convoluted the nephron is? Over here is kind of twisting on itself. Here I'm showing you an unfolded nephron. So keep in mind they're very twisty over here, and I'll show you later on how they look normally, how they look when they fold on itself. But here is I unfolded it to show you the different segments or the different sections of the nephron. So you need to know these parts of the nephron. So the first part of the nephron is going to be the Bowman's capsule. So the Bowman's capsule is this hollow ball, right, which is the first part of the nephron. And this ball-like structure, inside of it, it has the glomerulus. And the glomerulus, which is here in red, it's a network of capillaries. Okay, so remember we studied capillaries. Those are the exchange blood vessels. These are, it's a network of those capillaries for exchanging materials with the nephron. Okay, so the Bowman's capsule is part of the nephron. The glomerulus is part of the circulatory system. Together, Bowman's capsule and glomerulus make the renal corpuscle. Okay. The next segment of the nephron is part of the renal tubule. So we have here the proximal convoluted tubule. You can just call it proximal tubule. That's fine. So that's the first part of the renal tubule connected to the Bowman's capsule. We have that proximal tubule. Then we have the loop of Henle, which is this U-shaped structure here. And the loop of Henle is divided in 3 segments. We have the descending limb of the loop of Henle, the thin ascending limb of the loop of Henle, and the thick ascending limb of the loop of Henle. For the purpose of this class, we're just going to call it descending limb of the loop of Henle and ascending limb of the loop of Henle. After the loop of Henle, we have the distal convoluted tubule. And you can just call it distal tubule. And the distal tubule of several nephrons empty into the collecting duct. So this distal tubule empties here, another distal tubule empties here, another distal tubule empties here. And the collecting duct is going to take that to the renal pelvis. From the renal pelvis, as you saw right here, it goes out the ureter to the bladder. Okay? Why do I show you the different parts of the nephron? Because these different parts of the nephron have different characteristics and different functions. All right? So we're going to look at the functions. The function of the nephron is to make urine. Okay? And to make urine, we need there are three processes. The first process in urine formation is going to be filtration. What is filtration? Filtration is the movement of fluid with some solutes, right, from the blood in that glomerulus into the Bowman's capsule, into the lumen of the Bowman's capsule. Okay? So filtration only happens in the renal corpuscle, between the glomerulus and the Bowman's capsule. That's the only place where filtration occurs. And I'll talk about filtration in detail in just a minute. Then we have reabsorption. So in the remaining segments of the nephron, so all of the renal tubule, we're going to have reabsorption. And reabsorption is basically recycling things that got filtered, but we don't want to excrete. Remember, now we're in the lumen of that nephron. This is considered the outside of the body, okay? So if we don't want to waste that and excrete it, we're going to recycle it. For example, glucose. Glucose gets filtered. We don't want to waste glucose. That's the golden fuel, right? So we recycle that, we reabsorb that glucose. Where do we move it to? We move it back to the circulatory system. So here you see that the glomerulus has blood that comes from the afferent arteriole. So there are a number of blood vessels, as you see right here, right? This renal artery is going to come in and it's going to become arterioles, afferent arterioles. Those afferent arterioles become or end up in the glomerulus, which are capillaries. That blood from the glomerulus then goes out through an efferent arteriole, and that efferent arteriole is going to become a peritubular capillary. And so there are going to be peritubular capillaries associated with all the segments of the nephron. You're going to have a peritubular capillary next to each segment of the nephron throughout the whole nephron. Why? Because we need to be able to exchange materials. So once we reabsorb materials, that reabsorbed material is going to move from the lumen of that nephron into that peritubular capillary. It's brought back into the circulatory system, meaning it's brought back into the body. The last process is secretion. And so secretion happens again in all of the renal tubules, so proximal tubule, loop of Henle, distal tubule collecting duct. And secretion is when we have selected molecules that are moved from the blood into the lumen of that nephron. So things that didn't get filtered but we want to get rid of are going to be moved back from the blood into the nephron. So what is the amount of solute excreted is going to be what we filtered minus what got reabsorbed, right? Because that got removed from the lumen of the nephron, plus what we secreted while we added at the end. That's going to give you the amount of solute excreted. Any questions so far? Okay, I'm going to talk about filtration. All right, so filtration happens again only in the renal corpuscle. Renal corpuscle is going to be the Bowman's capsule and the glomerulus. Okay? And blood is filtered from the glomerular capillaries into the Bowman's capsule, and we make filtrate. So this depends on blood pressure. Okay, and let me explain how that works. You're going to have some blood coming here through the afferent arteriole into the glomerulus, and that blood is going to exert a hydrostatic pressure. Okay, so that volume of blood is going to push against the walls of those capillaries, okay, and generates this capillary pressure. So this PC here is capillary pressure. And let me get my pen here. So this right here is capillary pressure. Which is really a hydrostatic pressure. And it's going to depend on blood pressure. Okay, so the higher the blood pressure, or the higher the volume of blood going in here, right, the higher the capillary pressure. And imagine this glomerulus like a garden hose. You guys know garden hoses? You know, you have your hose connected to a faucet, right, like a water faucet, right? And then the garden hose, you have your plants here, and the garden hose has holes. Have you guys seen that, garden hoses? Yeah? Okay. If I turn on that faucet of water a little bit, then water is going to go into that hose, and that water is going to push against the walls of the hose, right? Because we have those little holes, then water is going to start moving out. It's going to filter from the hose onto your garden. If I open the faucet even more water is going to move into that hose, generating a larger hydrostatic pressure. Is going to push against the walls of that hose much more forcefully. Remember, pressure is force over area. You have the same area, but now you have more fluid exerting a force. So that increases the pressure, right? So more water is going to filter out that hose. Does that make sense? Same idea here. The more volume of blood I bring into the glomerulus, the more hydrostatic pressure, capillary pressure, the more is going to leak out. These capillaries are very permeable, so more is going to filter into the Bowman's capsule. But those holes are a certain size, right? So they're going to allow the fluid, which is plasma, which is basically water, 93% water, and some small solutes can move, okay? Even slightly bigger ones, like glucose, can move and get filtered, move from the glomerulus into the Bowman's capsule. But your gigantic red blood cells cannot move. Big proteins like albumin, which are it's a protein in the blood, cannot move, cannot cross. So you are moving some solutes, and you are moving some water, a ton of water actually, into that Bowman's capsule. But the big blood proteins like albumin, the red blood cells, they cannot cross because they're too big for those holes that you have in that garden hose, in that glomerular capillary. So the filtrate ends up being very similar to plasma, but not exactly like plasma, because it's not going to have all those big proteins. It's not going to have the cells that you find in blood, okay? But the osmolarity is going to be the same because water can cross, right? So 300 milliosmolar here, 300 milliosmolar here. Coming back to Rob's question about eventually having that osmolarity equilibrate, okay? So you're going to have that force moving, that filtering small solutes and that fluid into the Bowman's capsule. There are some forces opposing it, okay? What opposes it? Well, you have a filtrate now. You have fluid in that Bowman's capsule, so that's also going to produce a hydrostatic pressure moving fluid back into the glomerulus. And you have this osmotic pressure here. So I told you that the big red blood cells cannot cross, and the big albumin and other big proteins cannot cross. Those are solutes that are going to generate an osmotic pressure, trying to suck water back in. So these two forces are going to oppose the capillary pressure, which is related to blood pressure. But if your blood pressure is normal, right, that capillary pressure is going to be larger than the forces opposing it. And so you always have net filtration. Okay? If your kidneys are not filtering all the time, that could cause damage. Okay? That's when you end up in trouble. So there should always be a filtration rate, is what I'm trying to say. Okay? It can be lower or higher depending on how well hydrated you are and how, mostly hydration, and what your blood pressure is, but there should always be filtration happening in the renal corpuscle. What do we filter? We filter a ton of stuff. We filter water. I should give you guys the code. The code is 445. We filter water. We filter salts like sodium chloride, potassium chloride, magnesium chloride, calcium chloride. Filter bicarbonate and hydrogen ions, urea, glucose, amino acids. What's not filtered are the big cells and the plasma proteins. Water can cross freely, so that's going to maintain the osmolarity the same. So the osmolarity is going to be 300 millismolar in plasma, in the glomerulus, and in the filtrate, in the Bowman's capsule. The glomerular filtration rate is then the amount of filtrate produced by each kidney per minute. So the GFR is in mils per minute, how much volume of filtrate is produced by each kidney every minute, and so the GFR is 125 mils per minute in a well-hydrated, healthy human. Okay. Now I have a clicker question here for you guys, and it was for the code, but I gave you the code before. Okay, so how is GFR, how is that glomerular filtration rate regulated? So how much we filter, it's going to depend on blood pressure, okay? So here I have my afferent arteriole, which is equivalent to here. I have the glomerulus or glomerular capillary, which is this one right here. And then I have the efferent arteriole that joins with the peritubular capillaries over here. So this is the efferent arteriole that will become a peritubular capillary right here. And my question here is, what would happen to the glomerular filtration rate, or GFR, if the afferent arteriole vasoconstricts? Remember, arterioles, that's the thing they do. They're resistance blood vessels, right? They contract the smooth muscle, vasoconstriction, decreasing the lumen, increasing resistance, decreasing blood flow. Or they vasodilate. They relax that smooth muscle, making the lumen bigger, decreasing resistance, right? Increasing blood flow. So my question to you is, what happens if I vasoconstrict the afferent arteriole, the one bringing blood to the glomerulus, but the efferent arteriole remains the same, does not change? What would happen to GFR? Will it increase, decrease, or not change? What do you guys think? Remember that glomerular filtration rate, that filtration is going to depend on the capillary pressure here. And capillary pressure is a hydrostatic pressure, so it's going to depend on blood volume entering that glomerulus and leaving that glomerulus. So what do you guys think? Increase, decrease, or not change? Decrease. That's the correct answer. So let's look at why. If we have vasoconstriction of the afferent arteriole, we're going to make that lumen small and increase resistance, right? So it's going to be harder for blood to enter that glomerulus, right? We're decreasing the radius, more resistance to blood flow. We're going to decrease blood flow. Less blood comes into the glomerulus. So the blood volume in that glomerulus is going to decrease, okay? But the same amount is leaving. We haven't changed that efferent arterial radius. So we have less blood coming in, but the same amount leaving. We're going to end up with a smaller volume of blood in that glomerulus. And if we have lower blood volume in that glomerulus, there's going to be less push against the walls of that glomerulus. My garden hose, right? It's going to have less push. So then less capillary pressure, and that's the force for filtration. So then less filtration. Okay? Does that make sense? What about if I keep the afferent arterial radius the same? So I don't change afferent arterial, but I vasodilate the efferent arteriole. What do you guys think would happen to the GFR? Increase, decrease, or not change? What's that? You think no change? Anybody else? So let's think about what's going on here. I'm bringing the same amount of blood into the glomerulus, but more is leaving. So 10 comes in, but now instead of 8 coming out, it's going to be, I don't know, the same 10 comes in, instead of 8 going out, 10 leaves. Do I end up with more or less volume in the glomerulus? More blood volume or less? Less blood volume, right? The same amount comes in, but now a ton of it leaves. So that blood volume in the glomerulus decreases. Everybody with me? If blood volume decreases, what happens to capillary pressure in the glomerulus? What happens to that hydrostatic pressure? If I have less blood volume, do I have more push against the walls or less push? Less push, right? So capillary pressure decreases. That causes a decrease in GFR. Does that make sense, guys? So same amount is coming in, but more is leaving, more than it should, because this is vasodilated. So the resistance here decreases. If resistance here decreases, there's more blood flow. So more blood flow leaving the glomerulus, but the same blood flow as before coming in. So you end up with lower blood volume here, which decreases the capillary pressure. And if we decrease the capillary pressure, that's the driving force for filtration, then filtration decreases. Expect questions like this in the final, okay? I'll let you guys go and we'll continue with the rest later on. Guys, remember there is an assignment. Oh, by the way, before everybody leaves, no, I'll tell you on Friday. I'll have a surprise for the weekend. It's a good one. Yeah.